are listening to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Bannattini. In this podcast, I interview scientists and discuss their work, as well as the latest advancements and challenges in the field of brain mapping. Today, I had a great discussion with Dr. Randy McIntosh. Dr. McIntosh has been a scientist at the Rotman Research Institute of Baycrest Center at the University of Toronto since 1994. And since the start of 2022, is the new director of the Simon Fraser University Institute for Neuroscience and Neurotechnology in Burnaby, British Columbia, just outside of Vancouver. Randy obtained his Ph.D. in 1992 from the University of Texas at Austin in Psychology and Neuroscience, and did a postdoc at the NIH with Barry Horwitz until 1994. His group uses neuroimaging and computational modeling to understand the dynamics of healthy brains, as well as those from many different clinical populations, lending insight and providing potential biomarkers through comparing his dynamic brain models with empirical data. He's also part of an international consortium called TheVirtualBrain, which is an open science neuroinformatics platform for modeling the brain. Along with the exciting news of Randy's new position, he also just published a two-part book called A Complex Journey, which is a sci-fi novel that delves into the complexity of the brain. In this discussion, we talk about his research and specifically about this ambitious yet increasingly impactful project involving the virtual brain. We also delve into the different kinds of brain modeling approaches and what these different models provide. Lastly, we talk about his new position, as well as the Institute's unique goals of more effectively translating neuroscience to all-inclusive clinical care for individuals. I hope you enjoy this conversation as much as I did. All right, Randy, thanks for joining us for this podcast. And I really appreciate you spending the time to talk. And, we already had the introductions, and overall, I think that your impact on the field, I've always been aware of sort of what you've been doing, and I've sort of finally have delved into it deeply in the preparation for the podcast, but it's truly impressive. So why don't we just get started with just very quickly some of your background and, what inspired you? So what first inspired you to research the brain?
So, I've been interested in science as far back as I can remember, honestly. And I know it sounds kind of corny, but as far as I can remember, even or at least remember childhood, I was always interested in doing science of some sort. I got interested in psychology actually in high school. They had an elective course in my senior year that was basically psychology. And I thought, this is kind of cool. So we went through sort of, the overall theories of cognitive function and so on. But the motivation for the brain actually came in my first year in university with a class taught by Rob Sutherland at University of Lethbridge. At that point, Rob was a junior faculty at University of Lethbridge, and he was given the honor of teaching intro psychology to grads. And of course, Rob being a neuroscientist, he had a focus in terms of how he presented the course, and his course really focused a lot on the biological determinants of psychological processes. And I was just blown away by that. I said, wow, we can do this. And then the second course was actually from Ian Wishaw, who is well known as well in sort of behavior neuroscience, but also from his work in neuropsychology. And the course with Ian Wishaw was just blew me away. It's like, this is what I want to do for science, and that kind of just opened up the watershed, and I just kind of stuck with it, and ended up moving to Calgary, et cetera, and then the rest, as they say, is history.
Yeah, no, and so your PhD was in psychology and neuroscience, but now what you're doing though is it's sort of like it's very deeply modeling, and so to you probably learned that along the way, but how did you decide to get more into the modeling aspect, and what skills what did you have to take extra classes, or did you just learn along the way?
Sure. It was a bit of both. When I was I can't remember if it was in my undergraduate year or in the first couple of years of my master's degree in Calgary, but we were using a technique called 2-deoxyglucose autoradiography, which you probably know, was kind of like the predecessor for a lot of the neuroimaging that's being done now. And I was doing it in animals. And the one thing that impressed me about the technique compared to single unit recording was I could get the entire brain. And at that point, I was also heavily into anatomy. So I was reading a lot of literature on different systems, auditory system, limbic system, and so on. And I thought, well, this is fantastic because I can actually measure the entire system. But I can't see how they're working together. Because the analytic techniques are basically doing like very simple univariate tests, but I says we need to find ways to try and merge these data. So I started playing around with things like principal components analysis, and actually in one of my first projects in my master's involved using structural equation modeling or path analysis on 2DG data, and I was taking some multivariate stats courses as well to learn how to use these techniques. So it was sort of came complementary. Yeah. But I think the motivation for this was actually there was questions I needed to answer about the brain, and I didn't have the armamentarium, if you will, from my training, formal training. So I ended up having to seek other ways of doing that. And I had a lot of really good mentors in techniques like structural equation modeling. That were not part of sort of the traditional graduate students courses. So I got a bit of supplementary training from that. And the rest of it was just learn by doing, really.
Yeah. And that's actually right. I mean, I think that, a lot of people sort of gravitate towards that in some sense. And I almost feel like a lot of the people who do some of the most insightful modeling do start off from the side of neuroscience, and they just want to make sense of the data. And so they learn along the way, as opposed to people who start out as computer scientists. Certainly there's a lot of good modeling done with that, but it seems that, there's less of maybe a desire to really get into the neuroscience with the modeling in some sense. So starting from the neuroscience and learning the technical side is, I think it's a great, obviously, combination.
Yeah, it's tough because, the challenge with a psychology degree is that the maths that are underlying, especially the modeling we're doing now, you don't get that in psychology degrees. Yeah. So it's a formidable hill to climb, but I have good collaborators that can tell me when I'm going astray. So.
Yeah. That's right. I mean, as fields become more interdisciplinary anyway, it's expected that you're sort of supplementing your knowledge, along the way. So you had, for sure. So your advisor for your PhD work was Francisco Gonzalez Lima, and then of course, I know Barry Horwitz at the NIH, and you worked with him a little bit there too. How was that?
So when I first contacted Paco Gonzalez Lima, I was just finishing up my master's degree, and I was looking to do a PhD. So in the old days, you did a master's and you did a PhD. So I sent him a letter, actually a handwritten letter. I think it was handwritten. I can't do handwriting anymore, of course. But I sent him the letter and said, "Look, I've been doing 2DG with my master's advisor. You're doing some great work. Are you taking any students?" And he just happened to be starting up a lab. So having somebody come in the lab who already knew the technique was for him a benefit. So when I got down there, I was impressed because he had the kind of anatomical knowledge that really sort of pushed the envelope, because he knew the details of anatomy much better than most people I knew at that point in time, because he actually has legacies, goes back to almost to Hall. So he has that sort of tradition of very detailed anatomical knowledge. And we started just partnering, I think. So I went from being a student to a collaborator quite quickly, and essentially a lab manager with him, really. So he and I worked together in building the lab. I was quite involved in planning our move to UT Austin, which is where we ended up at some point. So I like that. He did provide a good framework for me, but he also allowed me to expand. That's really where the whole structural equation modeling went. It was really working with him, and that was how he supported that, because he saw that was a way to incorporate anatomy into the analyses. so there was that, and I think Paco also was very deliberate about making sure that I was making the right connections, networking, which is important for all people going through the system. Is who do you know and how do you make connections? Because it's a big field, a lot of people are busy. So he put me into talking to a lot of different people about this thing. Robert Satori is actually one of the first people I talked to about path analysis a long time ago. But it was interesting. It was at my very first Society for Neuroscience meeting in St. Louis. That's where I presented the path analysis stuff for the first time. And at that point, Barry was looking at the correlation stuff. So that's some of his first papers on doing the correlations of regions of interest in the brain started. And he was thinking about this technique, this path analysis or something like it. So he came by my poster because I sort of, I sent him a note saying I was doing this, he should come by. I think it was also handwritten.
Pre-email.
Yeah. You should come by and take this look at this. So he came by the poster, he looked at it, and he sort of went through it in great detail and saw about it. So, and then he came back and offered me a postdoc. And this is actually interesting because it was a good introduction to the NIH culture, which I'm sure you know very well. That he thought that rather than competing with me, he would be better for us to collaborate. And I think that was one thing that made Barry unique at NIH in some respects, that certainly when I was working there, the environment was extremely competitive. And there were collaborations, but the collaborations were usually done in terms of building little armies. So that you could, get the resources you needed to push your lab forward. But Barry was extremely open to collaboration. And he also was, he did a fantastic job in making sure that I had everything I needed to do the work. We all shared a very tiny office on the 10th floor of
I've seen that office.
Yeah. Should have a plaque on there somewhere because there was like 5 of us in an office that really should have had only one person in it. But anyway, I mean, the point being, I think, is that Barry was very supportive of me exploring things, and he and I, again, both Barry and Paco, I felt I was more. I learned from them in terms of their wisdom, but I think we also have moved together and we ended up being collaborators again. And in fact, that was true for both of those major mentors in my early career.
Cool. Yeah. No, Barry is. I think he just retired a little while ago, and
Berry Fest. I think just before the pandemic. I think the year before the pandemic, we had Berry Fest, which is quite a fun thing. On NIH campus.
The NIH. Not to talk about the NIH too much, but I think it's changed a little bit. I think that I always wondered about the dynamics that are set up, right? Because everyone has the resources, and it's like there's no motivation to push to collaborate. But I think we've started up core facilities, so people work through that. And
sure. I think that they established the neuroimaging. I think the growth of PET and MRI at NIH, I think, sort of broke down some of those barriers. And I do agree with you. It's a much different place now than it was when I was there and when you first got there, for sure. I'm sure. Yeah,
but it's still unique. It still has its own interesting idiosyncrasies and dynamics and things like that. But so you moved to Toronto in 1994, and one other. So then maybe just to begin talking about the virtual brain, which you've written many papers on many other things, but I think that it's interesting to see the evolution of the virtual brain, and now it's a prominent thing. It seems that your collaboration with Victor Jerza and Petra Ritter, I guess. So how did that? Because now when I see you at meetings, it's always there is this sort of sense that you're a team. You're working closely together. And how did that start and evolve in that regard?
Well, so there is this meeting called the Brain Connectivity Workshop, which started almost 20 years ago. The official first meeting was actually held by Rolf Cotter, who passed away 7 or 8 years ago, I think it is. And Rolf was a very, dyed-in-the-wool neuroanatomist as well, but he was the one pushing neuroinformatics. And so he and Carl Friston pulled together the first Brain Connectivity Workshop in Dusseldorf. The focus of that meeting was to really talk about the issues around how do we understand brain networks? And this is prior to the sort of network revolution, so it was an interesting time. So a lot of the recognizable names, like in the field, like Carl, myself, Michael Breakspear, Olaf Sporns, Giulio Tononi, all those people were sort of part of that group going forward. Yeah. And the second meeting we had in Cambridge, and that's where I first met Victor. And I gave my presentation, and he was probably the most pointed person in terms of asking questions that were kind of like, whoa, where's this coming from? And really pushing things, which I appreciated, because that was the nature of the meeting. The meeting is actually about discussions about the core concepts in brain connectivity. And I don't think it was at that meeting, but the subsequent one, he and I were chatting about, what's missing in this field, and we have a lot of really elegant work being done in imaging. That was sort of the beginning of diffusion tensor imaging. fMRI was sort of, pushing the field forward, and then we had all the, the graph modeling techniques. We had some of the effective and functional connectivity measures, and so on. They were kind of all floating around. And Victor and I were sort of sitting in a pub one day, and we were chatting about this at the BCW meeting. He said, we need something to pull us all together. We need something like a virtual brain. And I was like, oh, we can do that. So he and I started playing with sort of the primordial version of this, where I just gave him an adjacency matrix, a structural connectivity matrix, and then he started putting in dynamics, and all of a sudden we had a large-scale model of the primate brain, really not the human brain necessarily, was sort of an archetypal. But that opened up a whole host of opportunities. That happened in parallel with some funding from the McDonald Foundation that supported this network called Brain NRG, Brain Network Recovery Group. And that was a massive collaboration involving, Petra was on the team, Carl, Kathy Price, Giulio, Olaf. There was at one point 13 different universities that were all part of this group. And it went on for almost 10 years. Yeah. And that, to me, set both the theoretical foundation for things like the virtual brain, but also put in the opportunity to build the architecture. And the primary people in leading this were Victor and I initially, and then Petra was brought on because of her expertise actually in multimodal imaging initially, but also she's great in informatics as well. And it's just expanded from there. And I think it's filling a niche that was necessary to fill. And now I think it's become standard workflow in a lot of people's packages, and it's becoming part of international efforts like the Human Brain Project, eBrains, and so on and so forth. So it's done what I hoped it would do, and it's actually now giving us an opportunity to kind of carry things forward to the next level.
Yeah. So just starting from the top down in terms of what you said just triggered a question in my mind. So when you say the standard workflow, how do people right now, just before we get into the nitty gritty of it, what is their, right now, for instance, there's other people who do, Breakspear and Sporns, and they have their models, but in some sense, what you're doing is have a platform for inserting the elements of things that affect the dynamics, and then so their workflow would be sort of comparing it to their empirical data in some sense, or with the dynamics or the locations or interactions. So what would be a typical workflow if someone were working with the virtual brain?
So to back up 1 second and kind of give it maybe a perspective that could help, and then I'll tell you how it integrates. There is a difference between sort of just analytics. I'm not saying just analytics because I do the same thing, versus trying to build a model of what produced your data. And that's sort of the difference between virtual brain and other applications, is that we're trying to say what produced these data. Yeah. So it's building in an explicit theory that you can actually then see on paper. And that's a little bit different from doing graph modeling and so on. Where you do the analysis, and the analysis is the endpoint, whether that be looking at community structure, something like that, or doing something like PLS and looking at sort of some component space. But we're actually trying to generate these data. So that's, again, the generative versus discriminative or predictive modeling differentiation. So in a TVB workflow, what we might do, for instance, if we're looking at just resting state, we can get resting state data almost anywhere now. We can model it. So we get the diffusion data, we get the fMRI, and then look at, so what are the critical features that resulted in this particular pattern? And so you can speculate, well, maybe it's just the structural connectivity. So if we put a model that has sort of, vanilla dynamics and put the structural connectivity in it, does that generate the dynamics we see in the empirical data? Kinda. What else do we need? Well, we need time delays. Okay, put time delays in there. Oh, it gets actually much more interesting. Okay, what else do we need? We need noise. So you add noise, and all of a sudden you get a model that does really good. And you can do all the comparisons with appropriate nulls. So it becomes a way of an explicit testing of hypotheses about what you think are important features for generating those data. And that's a differentiation between a lot of the other approaches that are very sophisticated analyses, but the interpretation of whether the critical features for generating the data are a lot of times relegated to discussion sections, whereas in the generative model, that's part of the results. So it's a slight but important difference in how things like TVB, dynamic causal modeling is in the same line, that it's a generative approach. And that's the subtle but important differentiation. I hope that made sense.
No, that makes perfect sense. And so right, you might think, and just like you were talking about time delays, how important that is in somehow generating aspects of the noise. And even, you had a paper on looking at, for instance, entropy in the signal. And it's interesting trying to find ways of changing entropy with the virtual brain and seeing if that, it sort of suggests, it doesn't really, it's sort of an inverse problem because it could be a lot of things affecting the dynamics in some sense, but it suggests potentially certain types of, network, breakdown or whatever that might change with aging or Alzheimer's disease or things like that. Right. Interesting. Yeah, that's great. That's actually, that's a good, really nice framework for that, because I think that, when you talk about brain modeling, there's everything from, Carl Friston's free energy, which is not really even a model; it's sort of a construct to doing detailed network, like we said, sort of analytics, and then you just sort of talk in the discussion about what could be causing this sort of thing. And then there's also this other realm of doing like deep neural networks, and so people develop those, and their question is slightly different. It's not to understand the dynamics. It's sort of like to say, oh, this is how the brain does the computations. And it would be interesting. I mean, I was thinking about that in some sense, and that's a different question, but it would be interesting to integrate it somewhere. They should converge someday.
Sure. Yeah, it's a really, really, it's a good challenge to have, because I think what we're running into now is that the intersection between the sort of machine learning and biophysical based modeling approaches is happening. It's bound to happen and has to happen. Right now, Victor's been using a lot of the ML approaches to help with optimization of the biophysical models. But there are other things you can do with it, of course, as you said. I mean, if we try and reproduce, for example, the behavior that we see in a memory task, for instance, we have our fMRI data, we have the reaction time and accuracy measures. Okay, fine. So then we're trying to construct a multi-layered neural net that does the same thing. So what are the critical features in that we need to change to produce a memory deficit, for instance, instead of making assumptions? So it's again, it's a nice generative approach, but it's doing it at a slightly different level, which I think is an important one. But it's one that's a step away from the biophysical model. I'm not necessarily conflicting. Actually, can be complementary, because it's a way to integrate the cognitive domain that we don't have access to other than through some inference, like by reaction time and accuracy measures. So the gentleman, or colleague gentleman, he's a gentleman, Brendan Turner, who's doing some of this work trying to integrate the two different approaches. It's hard, but I think it's the way to go, because it allows us again to get that insight in terms of the cognitive processes that we can't see. We can see the results of the calculations, but we can't really measure them explicitly.
Yeah. Yeah, and so this is sort of trying to bridge that gap in some regard, right? Mm-hmm. So in that sense, it is unique. I mean, there's nothing like your construct, and it's sort of it's open access. You can download it, you can play with it, and I haven't seen anything. Certainly the Human Brain Project in Europe, they tried modeling individual neurons and looking at dynamics, and but this is sort of a different scale, and it's unique. It's completely unique, I think.
I think so. I mean, we built it to be I would say a service to the community, in the sense we're providing a platform for people to use to start testing their ideas, and it's purposefully, with one exception, it's model agnostic as well. So it allows you to use any kind of model you wish to look at the dynamics, either oscillators, or you can put in much more complicated multi-component biophysical models. And that's really up to the user to decide what they think is the most important set of dynamics to reproduce what they want to try and reproduce. And I think that's been helpful, because people can then use these. They don't have to use the virtual brain per se, but they can at least say, oh, we can do this. So they can write their own code to do that as well.
Yeah. And it does, we'll get into some of the clinical applications, but so just to talk a little bit about some of the architecture of it in some sense. So, so as far as the basic elements, I mean, you have so the brain divided up into network nodes, and it's sort of constrained by DTI data. And so that's the idea there, is that these are the white matter tracks, and that's where at least the highest rate communication, or the easiest communication, occurs. So do you, some people, it's interesting when people look at networks. They, one thought about DTI is that I think you're right. I mean, that's certainly that's where the super highways. But people always wonder. I think that when you have functional connectivity, you could actually have any sort of connectivity, but it's through that in that regard. And so that seems essential to looking at some of the dynamics as they manifest.
Sure.
So also, do you consider inhibitory or excitatory? It seems like when you have any oscillators or whatever, you have to consider some sort of inhibitory feedback.
Yeah. That's definitely it's either implicit in the oscillatory models, or explicit in something like, for example, the reduced Wang models in there. It has NMDA channels, it has GABA channels in there. Stefanescu, Ursa, a lot of those biophysical have excitatory inhibitory populations, and then tuning parameters for feedback inhibition to sort of dampen the range at which these populations can start spiking, essentially. So I said they're implicit or explicit, and depending on which model you use, again, you can make inferences by generating, for example, the currents for inhibition excitation, looking at the timing and the distribution of those, and then doing the generative model, forward model, see what does that look like now if I generate an EEG signal or bolt signal from that.
Yeah.
So you can bridge scales quite nicely, and Petra's group is leading a lot of work on that. It's working quite well. I was skeptical initially, but I'm happy that it's actually going in the direction that it needs to go.
Yeah, right. There's a lot of and actually, I'm really intrigued by that, because I think that with bold contrast, for instance, a lot of people think it's mostly excitatory, but then there's some groups suggesting that there's signatures of inhibition. Of course, people thought that the post undershoot, that still might be, or even habituation, like if you have a stimulus and then quickly in the visual cortex, it quickly becomes, damped, and that could be some sort of inhibitory. It would be really interesting to try to tweak that to see if you could mimic, some of the clear, bold habituation effects, to then go back to bold and say, oh, this part, is inhibitory, this part is excitatory, and maybe, make some more.
For sure. I mean, that does require a bit more detailed data as well, because some of the depending on the resolution you're getting from your scanner, a lot of those phenomena might not be visible, but they can be modeled, certainly. I mean, in some of Petra's, the first paper we published from Misha Scherner was the first author on that. We were able to reproduce that. It's well documented, this sort of negative relationship between alpha and the bold signal. Yeah. And when you take that alpha signature down to sort of its biophysical core, you see what's driving a lot of the oscillations is this on-off between the excitation and inhibition, like fast excitation and slow inhibition that sort of dampens the wave, and that results in your alpha frequency, and that's what you correlates very nicely with the decrease in the bold signal.
Oh, that's cool.
So it's a nice kind of connection, and it's again, I think that's the kind of direction I would like to see the field going more and more, because it allows us to sort of extricate ourselves from the quicksand around focusing on a single technique and saying that, this is how I think it works, and then going back to either your own experimental data or looking at even animal models to see, is this actually what happens or not?
Yeah, and that's a nice way to frame it, right? It's sort of a sounding board, against with a lot of techniques, and giving potential, a little bit deeper insight into what's happening, and then you go back and better interpret your own data in this regard, or even better use it clinically. So what would you say? So you mentioned, you've mentioned some in some of your papers the importance of. So there's two questions I have. One is the idea of the right spatial and temporal scale. I mean, certainly the brain is organized across many different spatial and temporal scales, and years ago there were like mean field estimations of what's important. It's sort of like is there a hope that, is there a worry that you're at at a scale where it's still an inverse problem and you don't really know what's causing these dynamics, or are you at the scale that you think that, right, you can make headway in understanding the empirical data, and you're somewhere in between, sort of making some inferences about the mechanisms? But I always wonder, there's many ways of producing a solution, of a dynamic solution. You ever worried that it's somehow you're missing some temporal scale or even some spatial scale that's either too small or too big, or whatnot?
Yeah, I mean, this kind of gets into, there's two aspects to your question. One is trying to build a model that's sufficient to cover what you think is important, but also the fact that this is a nonlinear dynamical system, or that's kind of redundant, but dynamical system, in which case it is by nature hard to predict. It has deterministic aspects, which is primarily the structural connectivity, but has a lot of stochastic aspects that are really, really sensitive to initial conditions. And that's okay, because that's the architecture is meant to be that way, so we can adapt. So then it becomes more a question of what is the minimal data set you need to provide some insight about the scale you're trying to measure. So, and that's the focus, right? So in none of the models that I can think of that we've done thus far in the TBB family globally, how we tried to make inferences about molecular aspects like synaptic events. They're population level inferences. And that's because we just don't have the detail. Do we need it? For the level that we're looking at, probably not. Because there's a question of, as you said, spatial and temporal scales. Some temporal scales are probably irrelevant to some other temporal scales.
Yeah.
So what happens at a synapse and the quantal release and so on probably has very little impact on the broad dynamics. And conversely, that's also true in the other direction.
Yeah.
But there might be, enslavement that happens, to use some of Herman Hochman's ideas, that you have sort of slow timescales that actually change the faster dynamics. And that's what we're interested in looking at, is does that interplay? And there we try and look at, can we have enough information between scales to look at fast and slow interactions? So the fast population dynamics, the slower network dynamics, and look at their interactions. And you can expand the model, certainly, to include more detail. Part of it becomes a question of model identifiability, which is a huge problem here, but also computational aspects. So as you make the model more veridical, it becomes a huge computational challenge. And even with the GPU systems they have in our system here in BC, and the system that Victor uses in Jülich, it could take several weeks to optimize your model. And that's just for one subject, never mind a population of 600 or 700. So there is sort of pragmatic aspects as well. But it's really a question: is the model you come up with at the end of the day useful? Does it actually guide you in terms of ideas about the brain? And then at some point you have to think about application. I think Victor's group and their work on epilepsy is a really good example of taking the focus on a particular scale and saying this is actually going to be useful for clinical intervention. And so they've now been able to put together a model called the epileptor that's now part of a national clinical trial in France for doing pre-surgical mapping, and it's working quite well. So it's evidence that these models are helpful beyond just knowing about the brain. They actually be helpful in a practical sense as well.
So is it a way? So just to jump to that really quickly, and I don't know, I haven't read up on it, but is it a way of? I mean, certainly with epilepsy, people have tried to use bold contrast just to look for unique correlations that are at the focal point. People have, EEG can kind of from the inverse. Trying to do some sort of calculation, you can sort of localize, but this does it better somehow by trying to model the entire thing, and then so it's a better inverse, a better local in that sense, or
it's looking for what are the likely what is or are the likely sites for the seizure focus, and what's the propagation zone look like. Okay. So you can work through the model fitting and then get Bayesian evidence estimations for which are the likely sources for the seizure focus, and then take that back to the clinician because they're ultimately responsible, obviously, and they can say, okay, that makes sense, and it's provided agreement with what the neurologist says, but also sometimes they find surprising results. It says, well, that's an interesting observation, and they find that TVB usually is providing them with pretty good guidance in terms of what decisions they have to make.
And that's even better than, for instance, let's say you have a person having seizures with an MEG, for instance. And I've always felt like fMRI had more potential for localizing seizure foci, but it's just, it's hard to get the particular population. You have to have them have seizures in the scanner, and or there's some critical seizures, but
right.
But this actually looks at just ongoing resting state. It doesn't assume.
No, sorry. It's using stereotaxic EEG.
Okay.
It does use DTI and sometimes fMRI and bold to inform as well. So it is trying to merge as much data as possible.
Yeah.
So it's saying that the hope is that we'll find out what is the minimum data set you need to acquire to actually get reasonable data, because these are stereotaxic EEG. It's not as invasive as sort of the intracranial stuff, but it is invasive.
Yeah.
And if we can do it with MEG or MRI, that makes the whole enterprise a bit less invasive and hopefully more precise ultimately. But the idea, and this is just getting back to the original point about why we want to use the virtual brain, is that there's probably no single data set right now is going to give us the answers we need. So let's find out how to identify the critical features in the data so we extract them more intelligently. So it's not just amplitude, it might be frequency, it might be other entropy, for example. That may be the key features, and we can model that and say this is a much better indicator of in this case a clinical condition.
Okay. Yeah, that's really cool. That's actually right. It's extremely practical, and it gives you concrete results in that regard. So what would you say, as far as the model is concerned right now, do you feel like it has any major gaps in terms of, what you would like to add to it? in the future, or?
Yeah, I mean, we talked about the deep learning aspects and the cognitive models. I mean, we've been quite successful in looking at resting state activity in terms of what the models suggest, and it does suggest that there's a really strong architectural determinism that produces these resting state dynamics. What we're missing is really the linkage to function in the sense of cognitive and behavioral function.
Yeah.
And that's what I think is an important next step, because we have now, again, the architecture to do a bit more detailed examination. I mean, we could, and we are, look at resting state models where we look at the various levels of the biophysical parameters and their relationship to like neuropsych measures and so on, like in the Human Connectome Project and so on.
Yeah.
But what I think we'd like to do is look at, can we measure ongoing cognitive function, getting back to the example of Brandon's Turner's work, and look at how the dynamics at that moment actually sculpt the emergence of the cognitive function that emerges from that.
Yes.
I think that'd be fantastic if we could do that, and that's really something that I'm very excited about doing. But it requires a bit more of thinking about how do we intersect the biophysical models with the cognitive models, and that's where people like Brandon are going to be quite important at pushing that.
Yeah, and it might be linked, but another paper that you have recently is in terms of looking at low-dimensional manifolds. So,
right.
Our behavior is sort of low-dimensional, and so you're trying to, if I understand it correctly, you're trying to, you have these incredibly complex dynamics, but you're trying to find this low-dimensional space of that somehow relates in some interesting way to the behavior. Is that?
Yeah. It's exactly in line with that, and this is something that again, Victor was he came up with the idea deriving from some of the work in synergistics that Herman Haaken had pushed forward. By the way, I'm just going to do a quick. If you want to see a fantastic talk, go onto YouTube and search for Herman Haaken and look up his talk he gave about a year ago on a video conference where he did a history of synergetics, but he did it on handwritten pages that he walked through. It's a beautiful, beautiful, beautiful talk.
What's his name again? Herman
Haaken, H-A-K-E-N.
K-E-N. Okay. Okay.
Yeah.
Herman Haaken on YouTube. And all right. History of synergetics.
And I think the important thing about his approach is that he's seen the evolution of the dynamical systems approaches to, like, he developed this idea of the synergetics. Scott Kelso worked with him on that, and Victor, of course. But it's actually linked to things like free energy and so on. So all these other ideas of maximum entry principle from I forgot the guy's name. Anyway, they're all derived from all these different principles, and they're all talking about the exploration of manifolds in a dynamical framework. And the manifold approach, in and of itself, its mathematical part of it is one thing, and people can explain it that way. Another way of thinking about it is if you can find the boundaries or the rules that constrain the dynamics of a system, that gives you more ability to get better insight in how that system behaves. Not so much being perfectly accurate in predicting it, but at least constraining the probability. So you can think about the manifold as actually putting the boundaries on a distribution of the possible configurations that a system can take. And that's going to be obviously across space and time. The nice thing about that framework is it allows you then to link up scales as well.
Yeah. Okay.
So we can then talk about going between, as I said, the biophysical and the behavioral scale, using things like these manifold interactions that can be in principle estimated. But it provides you with a way of kind of constraining the problem. So it goes from being, a hopelessly unsolvable inverse solution to something that at least gives you some ability to start integrating or inspecting the system at a level we couldn't do it before.
Yeah. Yeah. Yeah, no, I think right, and it seems like it has hope. I mean, it seems like, right, like for instance, I don't like, for instance, in resting state, the brain, is oscillating between multiple states, and people use ICA to pull that out in some regard. But actually understanding the manifolds and even trying to understand, the dwell time or the relative sequence of the states, or
Precisely.
You can get insights that way. That's really interesting. And that's sort of, you're right. It does constrain it. Like, it's funny. I keep on thinking of, once you look into the dynamics, it's sort of like the weather in some sense. I mean, we understand, the weather, but we don't have the, there are certain manifolds of the weather, but it's a little bit, that's even more somehow unconstrained because than in the brain, because you have the structure and
Well, it's an interesting connection you make there, because the Nobel Prize last year was for work in climate, in which dynamical systems modeling was central to actually making climate models, and that's essentially the same workflow we're talking about here. Is trying to come up with the critical features that we can estimate and derive the manifolds that constrain these trajectories. And that provides us, again, with the distribution that we can start exploring.
And then those themselves could be biomarkers in some regard. That are somehow more sensitive to see the manifolds than to actually just look at the data and try to pool it in some way.
Yeah, I'd like to think so. I mean, this is certainly one of my big efforts right now is to find ways to estimate these manifolds in a way that is useful for characterizing healthy function, but also in identifying trajectories that lead to dysfunction. As well. On
that note, we can just kind of quickly go into some of the clinical applications. I mean, it seems like, right, you mentioned epilepsy, stroke. I mean, there's some listings in your website looking at Alzheimer's disease, aging, even schizophrenia. And that, I was intrigued by that because looking at relative, excitatory inhibitory activity and inferring an unbalance of that. I don't know if you want to talk about any of those. I
mean, each one of them is probably a talk in and of itself. But one of I think one of the things that's coming up, and it's not going to sound terribly surprising until you actually dig deeper into it, is that almost every application we've seen thus far does involve a change in the balance of local and global dynamics. And that, I think, is a general principle of brain function that we need to really start thinking about seriously and embracing. So, we can talk about regional dysfunction and things like amnesia, where we've got maybe hippocampal damage, but the ensuing deficit that arises is not simply because that tissue is gone. It's also because of what happens to the brain afterwards. What does the brain try and do when this piece of tissue is missing? And that's this balance of local and global dynamics. So in schizophrenia, in even in epilepsy, and for that matter, stroke and dementia, there's a change in local balance, excitation inhibition. Now, the timing and so on is different between the diseases. There's probably differences in terms of the actual molecular components, but the manifestation is that there's a change in that balance. And the question then becomes, how does that balance get adjusted for from the global dynamics? Can it get adjusted for? If it does, then in theory you can adapt. If it doesn't, then you've got a cascade effect that might cause some clinical manifestation. Could be, delusions and schizophrenia. It could be seizures that propagate. It could be just an inability to integrate information because of degeneration, like an Alzheimer's disease. Because that balance is no longer maintained. And I'm not saying that that's going to be a solution for everything, but at least I think gives us a way of considering what a clinical manifestation means in terms of the brain dynamics that allows us to sort of balance our focus on region versus network.
Okay. So when you say you can imagine generalizing. So the whole idea of clinical manifestations is sort of like a reduction of your repertoire of behavior, and that's sort of could be right. So when you say local versus global, it's sort of like local, like specific areas that are lesion versus that the local activation and the network versus sort of like how it's connected to how it's more distally connected to other.
And what happens to those other regions? Because most of these deficits clinical deficits arise over time. So, schizophrenia is a great example, is that there could be things that happen very, very early on in life.
Yeah.
And, once the maturation of the system gets to the point where that dysmorphology or whatever is the underlying cause becomes critical, the network changes its dynamics. So even though it might be regionally focused, it's a network level problem. And I would, I could hypothesize that many clinical disorders are actually of that ilk. So it's a question. It becomes a question of, so how do we think about that in terms of how then do we intervene with the patient to try and improve their condition somehow? That's
a great way of, that's an interesting avenue of thinking about it. It brings to mind, I mean, there's a, just in general, there's another researcher that I know who studies brain energetics and the idea that, as you age, your mitochondria become less able to use glucose. And so the whole system is somehow reduced in terms of their metabolism. And then how does that break down? Like if you have a global reduction in metabolism, how does that bring down the network in some unique way? Or even how could it be manifest in terms of the entropy or right. I mean, there's all kinds of things that you think, oh yeah, that's bad. But then, you're giving insight based on your models.
Right. For you and I, we just keep on running. That way our brains stay healthy for a
while. That's right. Yeah. We both run and and actually speaking of running, I just, I remember one time running with you at a conference a long time ago in Brighton, I think it was.
Yes.
Yeah.
And Brighton on the beach.
Yeah. And I know for myself, I mean, that's just an interesting example of, my brain state after running is hugely different than before running, especially if I run in the end of the day or whatever. And it would be wonderful to try to model just that. And is it just simply increasing the metabolism, or is it some hormonal change, or, it's, that would be an interesting, I'm sure you've thought about doing that as well. So.
It's a question of getting the appropriate measurements, but certainly I have the same impression that I'm a different person when I come back from a run.
Yeah. Yeah. Right. I mean, a plug for running in terms of,
or even just walking for those who are going to listen to this, because just any kind of cardiovascular exercise, I think that's probably right now the safest bet for maintaining brain health. So,
I would 100% agree with that, but and it would be great to even show the evidence in your network models. There's so many more things we could talk about, clinical applications, but definitely, the holy grail of I mean, all these imaging techniques is that we want to find biomarkers, and I really think that, and so even with fMRI, so we can, there's certainly interesting insights about the brain, but we just don't have the, it's another question. We don't have the reproducibility potentially, or the power to pull out individual subjects, but it seems potentially, I mean, obviously people are trying, multivariate analysis, but it seems like what you're doing in trying to model this, it's a really interesting lever to sort of get at maybe increasing the signal to noise in some sense, sort of by, using this model as sort of a lever to sort of see these latent states, or to get at, potentially what's causing individual changes in dynamics. And so it might, I mean, it seems like there's hope for biomarker generation in that regard.
I think so. I mean, it's to use sort of a statistical analogy, by modeling these data, and especially modeling with respect to empirical data, it's kind of like you're extracting the most predictive features from the data that produce the what you think are the important characteristics of brain dynamics. And in doing so, it actually probably eliminates some of the noise in the measuring devices. So it's kind of like doing latent variable modeling, where you extract a latent variable and you have measurement error that gets eliminated in the course of modeling. We have some suggestion that that's true, like in our application to Alzheimer's disease. We improved the prediction quite a bit in terms of predicting cognitive status by using the model, compared to like just using functional connectivity or structural connectivity. and that, in my interpretation, was that we're actually extracting the critical aspects from the measured signal and then leaving the measurement noise behind.
Okay. Yeah, no, I think that's a really important point about your model as far as that's concerned. You're not, there's that improvement that you get potentially. It's adding insights to understand the brain, but also this very, very real clinical, because the clinical manifestations are all measurable. It's just a matter of putting it all together.
Yeah.
And you need that model to put it together.
I agree.
Just we can go on for so long, but I think I realize the time is starting to run short for. Just to sort of start to wrap up a little bit here, to talk a little bit about the future of brain modeling. So, certainly, just very quickly, where do you see it going, and do you think that it's just a matter of getting all the parts together and understanding the details, making more and more measures, more precise measures, or do you think we're missing something fundamental in terms of how it's being put together, some sort of principles that we can derive to help us increase our understanding?
That's a tough one. It's hard to predict the future in this game.
Yeah,
I'm sure,
especially breakthroughs.
Thinking about what would happen if we didn't have optogenetics, for example, in neuroscience, it would have been any different than where we are right now. I don't know. I think a lot of, and I'm going to sound very, dated in this case, like an old curmudgeon, but we get fascinated with the techniques. And forget about what the question is a lot of times.
Yeah.
And I think it is worthwhile thinking about, I don't want to pause the technology, but it's actually a question of thinking about what have we learned thus far, and what has provided us with that insight. So the technology is no question that we've learned new stuff, and fMRI certainly pushed us in new directions that would not have happened otherwise. And we're now going back to looking at other techniques that are complementary, like EEG, which has been around a little while longer, but is providing us a new perspective because of what fMRI is showing us. So to me, it's actually revisiting the questions. That's probably the most important thing right now. And thinking about what have we actually learned, and then how can we learn more. So I think integrating data sets is going to be a really important push. And then from that, in the clinical application, saying, okay, if we have the luxury of measuring every part of the person's body, can we then go and make this Uber model that includes all of their critical features, including, and this is something we're often missing, is their psychosocial and social cultural context. And from that, start deriving better indices of that person. And where that person sits in this broader distribution of for example, brain health. And we think about that in a different way now. We think about, okay, so this person is, the typical undergraduate at a Ivy School university. Their risk factors are quite different than somebody who's, the person who's working in a grocery store, who's the same age and doesn't have the same opportunities. So integrating those factors, I think, is going to be a change in the way we think about it, because we're not going to get much further along if we just continue to measure the Ivy School undergraduates. We have to think about a broader application, and then deriving from that, can we get measures that really matter to that person, that helps them. And that requires us, again, to pause for a second and think about what it is that we're trying to learn, and how can we improve that path to learning about ourselves, but learning about others, and then actually take that knowledge and put it in the hands of people who really need it. Okay. All right. So I think, there is definitely a push. You're absolutely right. Our standard pool is graduate students, and including a much broader spectrum can do nothing but lend more insight into, it all comes down to the individual, and there's similarities and differences. And we're just at the very tip of trying to understand
that interplay between what are the similarities, what are the differences, what are what's pathology, what's not, and
And I think that some of these things, just as a kind of a tangent. I mean, the measuring what matters beyond the brain, I think, is something that we as neuroscientists don't really have a good handle on. And that's where working with the people who are in the social aspects, talking to the philosophers, for example, who think about these things a lot, talking to people who are the clinicians in the front lines, even talking to the people who are affected, like going to the advocacy groups in developmental disorders, going to the Alzheimer's associations and saying, well, what is the critical thing? I said, well, it's not so much the memory problem, it's actually the navigation that's the issue, or the aggressive behaviors that are the issue. And this thing, so it's aggressive behaviors that are a problem. So can we actually get some insights in terms of what are the markers for that? Well, yeah, we can. We have this thing called entropy that we can use, and it measures the risk of delusional behavior in people who have Alzheimer's disease. And so that's the kind of thing that the dynamics, I think, that's going to be important for our field. And it's a huge area that can be explored and very, very ripe for the picking.
Yeah, and it seems that's having even information on your iPhone or whatever, just having something that, or even things that can track your behavior in that. So you're referring to that in some sense, like looking at the wider context, looking at every aspect of their social interactions, right? Like you said, whether they're aggressive, and how does that link to memory deficits? Yeah, I mean, getting a handle on that is so hard.
Well, we talked about complex systems, Peter, and this is a complex system, right? The humans and their environment and their social context is a complex system. And I don't want to digress too much from neuroscience, but what we're experiencing now with this COVID pandemic is a great example of how complex system principles have a tremendous effect if you ignore them. Like log growth functions, for example, and knowing what a log exponential growth looks like. It's like, what does that mean? No, it's not doing this, it's doing this. All of a sudden they go like, oh, that's a complex system.
And it's funny, we're evolved to not really, that doesn't come naturally to us to sort of comprehend that sort of thing. I completely agree. And there's questions beyond the horizon in terms of complex systems of idea space. Why do people feel strongly about things like whether they get a vaccination or not versus others? Certainly it's your peer group, but it seems like those are dynamics as well that we can maybe potentially get insight into. But
agreed.
No, this is great, and hopefully maybe you'll continue to expand beyond the brain and integrate this model in some sense, the virtual reality. Along those lines, okay, so that's a great way to that was a wonderful answer, and I totally agree. And actually, it gives me something more to think about in terms of getting a wider broad range of subjects. Looking at all the things that affect human behavior, more so than just simply, and how that feeds into brain dynamics in that regard. Because it may matter, it may not matter, but certainly there's more there that it's worth getting. So you have just a few more questions. You have a very creative side. You love music. You also you've just finished. Or just published two fictional books called A Complex, one book, but two parts, I guess, A Complex Journey, Brain Maze, and then The Next Day. Do you want to talk just quickly about that, just to sort of frame it and what you
it's almost as if we intended this conversation to go in that direction, because actually the motivation for the book is to give a bit more insights into complex systems. And it's done complex systems from the perspective of climate change. But there's an interaction with neuroscience in there. So when I stepped down from VP and director at Baycrest and did like a mini sabbatical. I was kind of thinking about what I wanted to do, and I was being pushed to write a book on complex systems from an academic perspective. And for the life of me, I just could not get the energy to do it. It's like, the books exist right now. I do not want to write another sort of academic text on that. So I was hemming and hawing. And then I had a dream where me and Victor and Petra were at a conference somewhere in Asia. And we went for a run after the session was done. And we ended up, and we actually were running in sort of like, army gear in some respects, like special ops gear. So we had like little guns in our backs, and we ended up chasing aliens into a sewer. And I woke up from this, and there was a very sort of vivid and elaborate narrative around the dream. And I woke up, and I was like, I have to write this down. So I went through and wrote the scene out, and the scene had sort of principles of complex systems, like emergence and multi-scale architectures that were sort of inherent in that particular scene. I wrote it down, sent it to Victor and Petra. They said, "This is fantastic." And Petra said, "This could be a book." And I went, "Wait a second, you're right." So all of a sudden, it's like I could do this. I could take this whole scenario and make it a science fiction novel about complex systems, about aliens who are influencing our climate, about using brain-computer interfaces to help find these aliens, and then help try and come up with solutions by modeling interactions. And that's all throughout the book, but it's done in sort of this sort of funny narrative. And it is a side of me, and I used to do creative writing in high school, like I think a lot of us probably did. And I kind of, I wouldn't say stifled it, but I kind of put it on the shelf. And then it sort of reemerged, and I got, the book was done in about a year, and just got published this month, I think, actually. And then I've got other short stories that are not really neuroscience things, but there is this creative aspect that I think is important. It's important for me. It's as soothing as a nice run, but I think it's an important complement to being a scientist to have those releases, because it does help us. It changes our perspective. And by changing our perspective in that moment, all of a sudden we see things in a different light. And you mentioned, and I said the same thing, like when you come back from a run, you're not the same person. And that's almost because you've all of a sudden, over that period of time, blood flow changes, brain networks change for a period of time. Yeah. And you've got a different perspective, and you feel fantastic. And that's exactly what happens when I write fiction or when I play my guitar.
Oh, that's cool. So it's sort of like, then I was just going to ask how you find time to do that, but in some ways, just like running, you make time, because it's pleasurable in itself.
Yeah. My wife kind of, she encourages it, because she says, you're miserable, go for a run.
Yeah. Oh, definitely. My wife does the same thing. She knows exactly when I haven't run. And, well, that's wonderful. And just one last thing, there was this it's on your website, sort of the concept of, I think it was Petra's, the virtual dream, where you actually hook people.
Oh yeah.
What is that? Just really quickly, that's looked fascinating.
Yeah, those ideas actually came out of a colleague who was in my lab for a number of years, Natasha Kovacevic. And she was primarily a programmer, but she also was an artist as well. And she was working on some of the early versions of the virtual brain with us, and I was kind of thinking, like, if I was to talk to my brain through a computer interface, what would it look like? And then so she started talking with Jessica Palmer, who's a graphics design artist, actually also does games, who's now working with Petra. And then Jessica and Natasha started thinking about, can we build sort of an art experience where you use, a low-res EEG. In this case, it's the Interaxon headset to drive a game, and the game is actually, basically using spectral power, but then using that to drive a series of animations that are sort of they're at that particular point is actually different kind of scenarios that were happening in animations that were projected onto a big screen, like a dream, but it was a collective dream, because we had, I think, 20 participants hooked up at the same time.
Oh, that's cool.
So it was a fantastic effort, because it did two things. One is, I mean, actually more than two, but I'll just think the two main things. One is it got people really engaged to think about, oh, this is cool. And this is neuroscience. And two, it is neuroscience because we got actually a paper out of it. We looked at, how can you learn and how fast do you learn and what are the determinants of learning? We had like 500 people go through this display in one evening. So we had a very large sample size, a good sample of men and women. We had some nice, data telling us like what are the determinants of how well people learn this. There's some different sex differences. So it's a nice way of engaging the public, getting them interested in neuroscience, but also being able to do neuroscience at the same time.
Yeah, that is so cool. You can imagine. I mean, I'm surprised it's already not. I mean, you already have the elements in terms of the gaming software. You just need a good high fidelity interface with EEG that you could actually, people could. You could market this. And certainly people, there is, certainly there's some biofeedback sort of devices out there, but it just seems like it's something that having a visual display could be interesting.
Yeah, I mean, Petra's group is working on that quite a bit. And if you go through Petra's lab website, there's a thing called Brain Modes that Jessica has been developing. That is an app. It's still, there's a lot of, it still needs some sandpaper to get rid of some of the rough edges, but it's working quite well.
Okay,
it's fun.
Cool. That's great. That's really, really cool. And how you would collapse the data and actually make it the most useful in terms of either feedback or just displays and whatever is an interesting question. Okay, finally, last question or last two questions. One, you're starting at as of January 1st, you're actually moving. You've been at Toronto so long, and you're starting a new position at Simon Fraser University, directing their Institute for Neuroscience and Neurotechnology, and that's in Vancouver. I guess it looks like it's right, in a suburb of Vancouver. So what motivated this, and what's your plans? I'm curious.
Yeah, and this is again, it's a nice segue from what we talked about earlier in the sense of inclusivity and expanding our focus. So SFU has a philosophy of inclusivity, and they feel like they have to be part of the community. So they embrace the notion that they're in the neighborhood, they're your neighbor. So it's like you can go to them for a cup of milk, for instance, if need be. And that perspective I thought was quite unique. I mean, and a lot of universities have that sort of in their mantra, but SFU seems to live that. And when I first started talking with them about this position, I thought it was like another neuroscience thing. But then as I started talking to the people, the VPs, the deans, the department chairs, and the scientists, and then actually what really got me moving was talking to the students, because they saw their work at SFU as being transformative in the sense of actually taking their knowledge and making a difference. So what I came up with, and we're still working on some of the details, is building an institute where the focus is to enable neuroscientists to do their work better. So providing them with the necessary support, like doing brain modeling, for instance, and give them like a part-time RA to help with that. Bringing together the neuroscientists across the SFU campus, but also more broadly, to think about burning questions like social determinants of brain health, and then finding the most effective means to get their information into the hands of people who need it. And that could be through technology, that could be through dissemination and interaction. So it's a more participatory motion towards neuroscience. And what I liked about that, that vision resonated because they have pieces in place to do that already. SFU does this. And doing that for brain, I think, is a fantastic opportunity. And it's like, I have to do this. So
wow, that's that is really cool. That's really, I mean, they have all the technology there. It seems like it's a really well-equipped center, but also at the same time this philosophy, and that's exciting and compelling, because there's not many places that, right, like you said, embody that philosophy, and I think it's important. It's a great move, I think, and I'm looking forward.
Yeah, thank you. It's been great running here too, Peter. So we've got some trails we can explore when you come visit.
Yeah, I know. Whenever I go to Vancouver, I just, one of my best runs in my entire life was in Stanley Park. There's this place, right? We had one of our meetings there, and I remember being up all night preparing the slides because I lost my slides, whatever. I just slept the whole day, but then I just went for a run. And it was like a 3-hour run. There's enough places to stop if you need food or whatever. And I just kept running. But I'm sure there's other places too. I mean, there's many. Vancouver is wonderful. It's
been fun to explore the trails as I'm thinking about the next evolution of virtual brain and so on. So.
Yeah,
or my next book, whatever.
It's a stunningly beautiful place. I love it. Well, good luck with that.
Thank you.
And at the very end, so is there any parting words, any advice anything you wanted to add, anything for advice for students or postdocs or perspectives on?
Yeah, I mean, I think the secret to my success, if you will, has been my collaborators and my colleagues. So I am not, there's no way I could have done any of this stuff on my own. And I think that's really important for students to realize, is that it's okay to collaborate. It's okay to have a network. It's okay to ask for help. And it's you co-mentor one another in doing that. And that's I think what's going to be critical for neuroscience going forward, is to think about it as a team sport.
That is great advice, and it's becoming more right. It's even more essential as we have bringing together different disciplines, and it's that's the only way you can actually, in terms of making progress. I mean, you can't have all the skill sets, and so you really do need help.
Yeah, definitely. Yeah.
Well, that's wonderful advice, and this has been a wonderful discussion. Thank you very much for coming on.
Thank you, Peter.
All right.
I appreciate it. Enjoyed it.
All right. Well, thank you. So did I.
Neurosalience is brought to you by the Organisation for Human Brain Mapping. This week's episode was produced by Anastasia Brovkin and Kevin Setek.